So let's continue here with phage typing. That's also a way in trying to sort of narrow down the type of bacterium that you're looking at and trying to classify it, because you're letting viruses, bacterioviruses, loose on that bacterium and see if a phage, which is a virus that infects bacteria, if that particular phage is able to infect that bacterium, and that will give you a better idea of what type of bacterium you are dealing with that you're trying to classify. So on a plate, you're looking for clearings. So basically the virus, the bacteriophage, has infected the bacterium, and now the bacterium is lysed. It's basically exploded, and now you, instead of seeing a lawn of bacteria on your plate, you see an area of clearing, and that area of clearing is called a plaque, because these bacterial cells were lysed by the virus. And so here, this is what something like that might be looking like. Phage typing here of a strain of Salmonella enterica bacteria, and you see these areas of clearing. Otherwise here, everywhere there's a haze here, except you can see here definitely an area of clearing where these bacteria, they can't grow anymore. They got infected by this virus, and they lysed, they don't exist anymore. Okay, fatty acid profiles. Not sure how often this is still done. I don't know much about it, and I'm not sure how common these fatty acid profiles are to classify bacteria. As far as I know, everybody has been sort of shifted over to just sequence the thing, do PCR on it. So, but here it is. FAME is the method. Fatty acid, methyl esters. They provide profiles that are constant for a particular species. And as I said, I'm not sure how common that still is. Flow cytometry is pretty common. That's basically a way where you are taking a sample and you are using a laser to sort out cells. So flow cytometry is a way of cell sorting, where you're using a laser beam to sort cells with similar characteristics in one test tube and sort others into a different characteristics in a different test tube. So you're sorting out cells. A cell sorting machine is that. And you're using a laser, and you're using fluorescence to sort out cells. So basically here is that's a pretty nice way of showing here. So this is fluorescent cell sorting. A lot of people call it a FACS sorter. So fluorescent activated cell sorter. Here's your mixture of different cells in this test tube, and then you're running it single file. These cells, they will run single file by a laser beam that's being shot on them, and then you're sorting out the cells into the ones that, I don't know, the brown bigger ones versus the little yellow tan ones. They go into this test tube. So that's a cell sorter, and it works pretty well. A lot of people do that when they want to get like white cell profiles, white blood cell profiles from a patient. You can tell, you can sort the white blood cells out, and you get a scattergram of the different populations. Okay. Now here is the big one, sorting out or classifying microorganisms based on their DNA. And that's widely popular, especially nowadays where we have these very efficient sequencers. So in a very broad categorization, you can just look at the percent guanine and cytosine. And so there's basically, for the gram positives, you can do the high GC versus the low GC species. That's pretty easily done. And then you can get definitely a more detailed profile by doing DNA fingerprinting, where you're taking the DNA of an organism and you're subjected to cutting with restriction enzymes, and then these restriction enzymes, they will cut, and the profile that you're getting after separating the different fragments will tell you how closely related certain species are. So it's a genetic fingerprint that you can do not just on prokaryotes but on eukaryotes. It's basically you're cutting the DNA of an organism with certain enzymes, and that gives you a profile of fragments, pieces of certain length, and this restriction fragment length polymorphism, that's what it's called, the RFLP, that will, if you then run these fragments, you separate them out by size, then this profile gives you this genetic fingerprint. And you can tell that, for example, Species 2 and 3 clearly have a similar profile, and then species 4 and 5 have a similar profile, and species 1 and 6 show a similar profile. So these profiles, these restriction fragment length polymorphisms, that's a very useful way of establishing genetic relatedness. So we call that genetic fingerprint. And this here is maybe the most popular method these days, the nucleic acid amplification test. That's a PCR reaction. So PCR stands for polymerase chain reaction, and it's a wildly popular method because it is effective, it is highly specific, it is fast, it is cheap, and there's really nothing negative to say about it. PCR, when it first came on site, it immediately just basically started to invade all biological labs. It's just wildly used everywhere. And it's also used in forensics. It has so many applications in diagnostics and even in forensic science and medical applications. It's outrageous for research purposes. So it used to be that each department had maybe one PCR machine when they first came out, and then every lab then pretty soon had one, and then every scientist had one, and then when I left research, pretty much everybody had at least one. Some people had multiple PCRs, and I did some PCR heavy test, developed some PCR tests myself, but the test that's used for COVID-19 right now, when you get a test done, that's an RT-PCR test because it's an RNA virus, and so you need to first reverse transcribe it, so RT, and then do the polymerase chain reaction to determine whether or not somebody's positive for COVID-19. But anyway, so back here, let me say a little bit more about PCR testing. So the PCR machine is a DNA copy machine, and you're taking a characteristic DNA sequence that you know is characteristic to a certain organism, and then if that sequence is present in a sample that you're testing, then the PCR machine will copy it, and with every round of the PCR machine you will duplicate. It's basically a replication of the DNA. So you're copying the DNA with every round of the PCR machine. You're doubling the amount of DNA. And after 30 or 40 rounds, you have a significant amount of DNA, if that particular sequence was present, that you can see then on electrophoresis samples. Okay, so here, nucleic acid hybridization, it's a little bit different. You're still separating out DNA pieces by size, but then you're coming in with a labeled probe, and you're looking for a matching sequence on the DNA pieces that you have separated by electrophoresis. So that's nucleic acid hybridization. Here's an example of that. So DNA hybridization. So you have two organisms, A and B here. You will heat them up to separate out the double strands, and then you will separate them by electrophoresis, and then you will go see if with a label probe you can find a matching sequence on usually a nitrocellulose or nylon membrane. So that's nucleic acid hybridization, otherwise known as southern blotting. So in southern blotting, you're using this nucleic acid hybridization to identify unknown DNA from a microorganism. And here it's maybe this image is a little bit better, shows nucleic acid hybridization, how it's done. So you have some DNA from an organism, you will extract it, you will separate it out on a electrophoresis, or you have colonies that are here then transferred onto nitrocellulose membranes. You will then have to break open these cells so that the DNA of the organism is exposed, and then you're coming in with a labeled probe. So you will come in with a DNA of known sequence that has either a radioactive label or fluorescent label, and here they have the fluorescent probe right here, and if it finds its match on the nitrocellulose, then it will get stuck there by hybridization. So because it's a matching sequence, it will form a double strand, and then you can determine by the fluorescent signal that you're getting that, oh yeah, my probe has found its match, so therefore that DNA that I was looking for was actually present. If you want to test for multiple sequences simultaneously, you can use DNA chips. That technology, when I left research, it just started up and it was fairly expensive. It's gotten a lot cheaper these days, but there you can test maybe basically doing PCR reactions simultaneously on a DNA chip of, let's say, 1,000, maybe up to 10,000 genes. And so these microarrays, that's pretty cool. You can detect then the presence of certain sequences by fluorescence, and here's what something like that might look like. It's basically doing multiple PCR and hybridization reactions with fluorescent probes simultaneously that will give you sort of a profile of different DNA sequences that are present or not present. And here's another DNA chip and what that might look like. So you guys don't get to try this out. We don't have that technology available, but it's nice to know that it exists. So DNA chip tests for multiple signals, or the presence of multiple sequences. And here you have quite a few on this chip right here. And gives you a pretty detailed profile of what type of DNA was present in a certain organism, in a certain sample. Here's another cool method, the fluorescent in situ hybridization. In situ means in the living cell, and so there, this method is abbreviated FISH for fluorescent in situ hybridization. And you're using fluorescent, again, fluorescence, the label DNA or RNA probes that then find their matching sequences in a living cell in this case. So that's in situ hybridization. And well, after you've done all your sequencing and your profiling and everything, most people are putting these days, they're putting their data into databases, and then you can actually, if you have an unknown organism and you've run some tests, preferably some genetic tests, and you know something about the sequence, and you can look for similar organisms that were already part of a database, and then you can establish your cladograms. In the old days, they used to do these dichotomous keys and sort of these classic microbiologists that were sort of establishing these phylogenetic trees. But dichotomous keys, I believe, are not that popular anymore. You just go straight into the databases, throw your sequence in there that you of your unknown microorganism, and see what it's related to. And so here is an example of what that could look like. That's our last slide here, I think. And here's a building sort of this cladogram of relatedness. And here, let's skip down here to the bottom. So where you're basically taking a look at sequence cell identity. Actually, this is slightly inaccurate here. This should say %. So either something is similar or it's not, or if you want to establish the identity, then you can do it by %. So % identity, this should say. I know you might think, oh, she's splitting here, but you know, I'm sorry, I can't stand this inaccuracy. So it should say % identity. And if you say, okay, here we have a bunch of lactobacilli. So Lactobacillus brevis, this particular San Franciscensis is 50%, has 50% sequence identity to Lactobacillus brevis of the species Acidophilus. So anyway, we're looking at particular genes or particular sequences, parts of a genome of a species here, and then you can establish your sequence identity by %, or you can say, so, well, they're similar to one another without specifying a particular percentage. But then here, we sort of have this relatedness of the species. And if you're looking at something like this, then you would say that For example, Lactobacillus sanfranciscensis is not that closely related to this Lactobacillus brevis, but definitely very highly related to Lactobacillus plantarum and so forth. So anyway, I think you get the idea, sort of this relatedness. A lot of people use the percent identity if they want to describe the relationship of species to one another using genetics. And that's it for this lecture. So thank you for listening.